Hello and welcome to Coco, where I'm going to teach you topics that you might find difficult. In this video, we're going to learn about enzymes. Most of it, the notes that I'm using are going to be based on CIE syllabus. So those of you who are doing AQA or Edexcel could also benefit from it. So let's begin. First thing we're going to cover is what are catalysts. Catalysts are anything or something that increases the rate of a reaction. When I say rate, I mean multiple times per second. Now, catalysts are substances that increases the rate of a reaction. Substances that increases. Mind you, these are notes that you could use in your exam papers and gain marks. This is short for reaction. Say you have something happening with you right now in a bowl, in a beaker, you have added something, but the reaction is taking forever and ever. So you put this substance called as catalyst suitable for that reaction. It will be faster in shorter time. Second thing about catalysts is that they do not get changed. Is not changed by the end of the reaction. At the end of the reaction. When you complete it with your reaction, you'll find that the substance is as new as it can be. It's like nothing had just happened. So it does not get changed. And this is a key point to write in your exams as well. It's called the additional mark point. Usually these questions come for 2 marks in CIE, IGCSE biology papers. Next, we're going to cover what are enzymes. Enzymes are the same as catalysts, but they are just biological catalyst. What does that mean? It means it is present, these substances are present in our bodies, in living organisms. And they are not any kind of rocks, they are protein substances in our body. So if a teacher asks you what are enzymes made of, you are sure to answer them protein. Alright, next we're going to cover what are metabolic reactions. What are they? Have you heard of a teacher using these words? Let's see. They are any chemical reactions. Again, I'm going to remind you, this is short for reactions taking place in our body or in living organism. All the time. You can write these down as notes and use it in your exam paper. These are mark scheme answers too. Now, so when I talk about enzymes, we need enzymes in these metabolic reactions that's happening in our bodies all the time. Usually they are really slow. Without enzyme, they would just go on forever and it's not enough to sustain our life. So we need it to be faster. That's why we have enzymes to speed up this metabolic reaction. Next, we're going to cover some examples of enzymes. Let's go for green. 1. We have an enzyme called amylase. Amylase, the ASE in the end. This one digests. Digest means breaking down. Digests starch to maltose. Simple sugar that's more simpler than starch. Alright? I'm just going to write here simple sugar. Another example of enzyme would be protease. Protease. This one digests proteins into its smaller version, which would be amino acids. Amino acids are the small bits that's broken down from proteins. Basically, proteins are made of amino acids. Next, third example I would give, the last one, would be a catalase. Do you notice how they end with the letters A, S, E? That's how enzymes are going to be named after what reactions they are acting as a catalyst. In a catalase, this guy breaks down H2O2. Wait. Now, H2O2 is hydrogen peroxide. They are made in our body as a toxic substances. They cannot stay in our body for longer. They are just dangerous and lethal. These guys needs to be get rid of as soon as possible. So wherever H2O2 is present, catalase would break hydrogen peroxide. Is that toxic? They will break these guys down to simpler, safe substances like water, H2O, and oxygen, O2. Alright, next we're going to cover. So I've told you that enzymes are found in our bodies and other living organisms. Right now I'm going to give you one such example where enzymes are also present. I would say, let's go with that. I'm avoiding red and green. Oh no, I did use green. Okay, whatever. Where do you find it? Okay. In seeds that are germinating, in the sense we have supplied water to it. So when they soak up water, we all know that seeds also contain stores of food for itself. So when they soak up the water, they have an enzyme inside them already called amylase. If you recall, amylase is the one that digests starch into simple sugar like maltose, or even simpler, glucose. So when water is there, amylase is activated. It gets activated and breaks down starch to, shall I add maltose or glucose? Let's add maltose. That's the accurate one. So inside the seed there is an embryo, the baby of the seed. This guy will use it, the embryo, of the seed uses it for providing energy. Glucose or simple sugars are used for providing energy. So they are using this energy for wait a minute, guys. For growth or making new cells so that it can multiply and become a shoot or a root. Alright. In this slide, I'm going to teach you the lock and key mechanism. You must have heard of it before. The lock and key mechanism is related to the enzymes. Basically, how an enzyme works. So an enzyme got a certain fixed shape. Got a certain fixed shape and allows another molecule, the ones that we're going to break down, another molecule to fit into it. I'm going to show you how. I hope my handwriting is not that messy. Now the thing is, it may look like this. Yeah, this may be our enzyme. Say protease. So it's going to break down protein. Our protein looks like this. So the shape should be an exact fit to fit right here for this enzyme to work. I hope you got the idea. Basically, the fit has to be perfect. The fit has to be perfect for the breakdown to take place. Breakdown to take place. Let me go back to the slide here. So in short, the enzyme is acting as a lock and the other molecule is acting as the key, the protein. For this to happen, the break to happen, we have a lock right here, and this shall be our key. In exams, when we want to describe this lock and key thing, we write a point that is the shape of enzyme and the shape of this substrate. I'm going to get back to what this means. Substrate are complementary to one another. To one another. Now I'm going to define you what a substrate and a product mean. When a protease digests proteins, the amino acid, we write it like this: protein, and this long arrow, and then right at the end, amino acid. Yes? And then above this arrow, we write the protease, because this guy is only helping to speed up the reaction. He is not part of the reaction or the reactant. Okay, so on the left hand side we have the protein, so we call this guy on the left hand side substrate, and the one on the right hand side we call it a product. On the next slide I'm going to define you those words. Substrate. Let me just erase that out. It is a substance on which the enzyme acts. The substance on which the enzyme acts. On the previous case, it's the protein. And the product would be the molecule produced. Next up, I'm going to explain two other reactions called anabolic reaction and catabolic reaction. This is also included in the syllabus. Anabolic reaction. This is a reaction in which large molecules are not broken down but built up from smaller molecules. You're probably confused because all this time I've been telling you that enzyme or catalyst actually breaks down stuff from big guys to small guys. Reverse could also happen in other catalysts. They can go from small molecules to big molecules, in the sense they're building it up. The next would be catabolic. This is the one that I've been explaining all this time, catabolic reaction. Reactions that split large molecules into smaller ones. Next up, we're going to learn the active site. What is an active site? Now, let's go back to the example of a protease. Let's take protease again. In a protease molecule, let me draw the protease enzyme. Yep, it looks like that. Not really, but this is the drawing I'm having. It has a dent. Can you see this dent right here? Dent. This dent here is called the active site. Enzyme has a dent called the active site. Now this shape, like I have shown you before, is complementary to the protein molecule only. Where is it? Right here. Like I told you, this guy is only for protein, probably this one for fats. You can see how this round curve will not fit in right here. It's only the protein that has these two edges. The fit has to be perfect, like I said earlier. So only when the fit is perfect, when the protein molecules fit right here into the enzyme, the breakdown will happen, protein gets broken down. Starch or fat molecules, they do not have the same complementary shape, they won't fit in and they won't get broken down. So for them to be broken down, you need to have another enzymes, like for example starch, you need amylase, and for fats you need lipase or lipids. Lipid is fat, enzyme for lipid is lipase. I'm going to show you a photograph I have borrowed from Google right here. This right here is the enzyme. They have shown you the active site, and this is a substrate. Like I said, it's probably one of those nutrient molecules coming in, and see how the fit is perfect. Now when the fit is happening, we call it an enzyme substrate complex. An enzyme substrate complex, it's for temporary time being while it's being broken down. And then from substrate we can see how the colors have changed. It has become products. This time around we use the word enzyme products complex. Usually I don't write this in the exam, I just write in the answer that an enzyme substrate complex is formed and then products are released. Like here, when we have the products, they unbind, they detach and they get released here. The products leaving an active site of an enzyme, and we have the tiny guys that we want, that are useful for our body. Well, I hope you understand what I explained over here so far. This is how simple I can go for those of you who have trouble understanding enzymes. And I think I missed one note. No, I did not. I definitely covered that enzymes are unchanged, catalysts are unchanged at the end of the reaction. Can you see how they look same as they were earlier? Let me show you. This is same shape. Same shape. They did not change. Can be used over and over again.